What's up guys, BIOBEAN here. So today we're going to be talking about some mutations and how they're introduced into the DNA. So let's get started. If you've just come from my video talking about the Human Genome Project, you know that we've been able to sequence the entire human genome and we know what the sequence contained within it. But we also know that there is a lot of variation in genes from person to person. Some people have blue eyes, some people have brown eyes, some people are more susceptible to certain diseases and some aren't. And so there's actually only 2% of the human genome that varies from person to person. So all 98% of your other genome is identical from you to someone that's in a far distant country. And so this 2% of variation mainly comes in the form of single nucleotide polymorphisms. So it's kind of a long word, so let's get into what that is. Single nucleotide polymorphisms, or better known as SNPs, are the most common type of genetic variation among people. So let's break down what that means. So single means one, nucleotide are the bases that make up our DNA, and polymorphism is a change within it. So a single base change within our DNA is the largest source of variation. So that means in this particular location, on this particular chromosome, you might have a G, someone else might have a T, someone else might have an A, and so there's variation in that particular nucleotide. These SNPs can occur anywhere within the genome, but they're most frequently seen in spaces between genes. This is because if you had changes within a gene, that might then affect your ability to function as a human being. And so most variations exist in regulatory elements or stretches of unknown DNA where they cannot affect your functioning as a person. However, some do have effect on your health and development. Some SNPs actually are associated with particular health and diseases. Frequently, people will do DNA screenings before they have kids in order to assess their susceptibility to particular diseases and their potential to pass these on to their offspring. These single nucleotide polymorphisms actually have an effect on our susceptibility to a disease. They can predict an individual's response to certain drugs. They may predict how you respond to certain environmental factors and toxins. They may tell you your risk of developing particular diseases. And our knowledge of these SNPs has really improved our ability to provide healthcare to individuals because we are now able to sequence their DNA thanks to the Human Genome Project, and then be able to tell based on what particular SNPs they have, which drugs would be most responsive to that individual. So that was a little bit into why SNPs are important and what they are. Let's get a little bit into what they look like. These SNPs have different effects based on where they fall within the genome. So I told you that many SNPs do not actually fall within genes because that might affect the functionality of that particular gene. So if these SNPs fall within spots that are not genes, generally they don't have any effect that's detectable. But if they fall into genes, then their effect depends on where in the gene they were changed. This is because of codons. If you think about the way that DNA is arranged, there's only 4 nitrogenous bases. There's the A, the T, the C, and the G. But those 4 bases have to then code for a protein, and that protein is made up of amino acids, and there's 20 different amino acids. So there has to be some way to go from only 4 nucleotides to somehow make 20 different amino acids. This is done through codons. Codons are essentially a unit of 3 nitrogenous bases. So in this string of DNA here, there are 3 codons present. The polymerase is going to read this and copy it into mRNA, and that mRNA is then going to be translated into a protein. And as it translates it into a protein, it reads each codon. So it doesn't look at what each individual nucleotide is, it looks at what the set of 3 nucleotides are. Here in this stretch of DNA, we have 3 codons comprised of 3 nucleotides. And when this goes to be translated into a protein, the ribosome is going to read each unit of 3. So it's going to read each set of 3 nucleotides, and that'll tell it what amino acid to put. So every codon has a corresponding amino acid that it codes for. Kind of think of this as binary for computers. So there's a string of 1s and 0s, and that tells the computer to store something over here or over here. When we're talking about SNPs and their effect on genes and codons, it's important to also understand the Wobble Hypothesis. So this is the hypothesis that the first two letters of the codon are the most important. And generally speaking, the last nucleotide in the codon, it does not change what amino acid is present. So here you can see 4 different codon pairs. They all start with the same 2 nucleotides. They're all starting with that CG, and they differ in their last nucleotide. However, they're all coding for the amino acid arginine. Just a quick note here, you're seeing the U's instead of T's because the codons are generally expressed as mRNA because the codons are what's being read to create a protein. So the codons are read at the mRNA level where they are translated by a ribosome into a amino acid. So wherever you're seeing a U here, it tells you you're at the mRNA level, and if we were at the DNA level, there would be a T. So this is the Wobble hypothesis in effect. So no matter what that third nucleotide is here in this codon, you are always going to get arginine. So let's talk about how this relates to SNPs. All right, so here we're looking at a table of possible SNPs that might occur inside a gene, and we're going to utilize our knowledge of codons and the Wobble hypothesis kind of decipher it. So here, looking at the very left side, we have the 3 different levels. So we always start at the DNA level, and that's our basic set of instructions for our protein. Then that gets transcribed into mRNA. That is why you're going to see some places where there should be a T, there's going to be a U, because that's how we know we're in our mRNA, is there's going to be that uracil instead of thymine. And then we're at the protein level. So mRNA is always translated into protein, where that then goes on to do functions. So we're looking at the DNA, we're looking at the codons, we're going to see what that translates to into mRNA, and then we're going to see what type of amino acid that is then going to tell the ribosome to put on the growing protein. So here is our original no mutation codon. So we have our TTC. So when that gets transcribed, that is going to be transcribed into its complementary mRNA. So TTC will be transcribed into AAG, and that is going to make the amino acid lysine. So now let's look what happens if there is a silent mutation. So a silent mutation is going to be a SNP that occurs in a spot that will not affect what type of amino acid is going to be placed there. So here, let's look at this silent mutation. So our standard TTC codon is getting converted into a TTT. So that C is converted into a thymine. So that was a type of single nucleotide polymorphism, is a conversion from a C to a T. So when this codon gets transcribed, it is going to be converted from its DNA form of TTT to its complementary mRNA, which is going to be AAA. So we're following through the mutation highlighted in red. So now this AAA codon is going to be translated into the amino acid lysine. So we see here that there was no actual change on the protein level, even though there was a change on the DNA level. This was because of the Wobble hypothesis. So because it was only that third letter on the codon that was changed, it ended up still coding for that same amino acid. So there was no actual change in what we saw on the protein level, so we call it a silent mutation because it doesn't actually have an effect on the protein that's going to be produced from that gene. So then on to the next one. So we see here, this has the first nucleotide change within this codon. So looking to our original TTC codon, it has been changed to ATC. So that first nucleotide is now changed from a T to an A. So that ATC is going to be transcribed into a UAG. Again, that we're looking at the complementary. So the A normally would be a T as its complementary, but because we're in our mRNA, it's going to be a U. And then this UAG gets translated into a stop codon. So this would tell the ribosome, this is the end of the protein, stop going here. So if this was the beginning of a protein, you might only have 2 amino acids added before you reach this stop codon. So this is what we call a nonsense mutation. So anytime that a change in the codon results in a stop codon, we call that a nonsense mutation, because now it will essentially make a nonsense protein because it doesn't have everything that it needs. It stops well before, or even a little bit before, the end of the protein should actually be. So anytime that a point mutation results in a stop codon, we call that a nonsense mutation. And then on to our last type of SNP or point mutation is our missense. So again, looking at our standard, it should be a TTC, but in this case, it is going to change into a TCC. So now we have that middle nucleotide of the codon changed. So when we get transcribed into our mRNA, it is going to convert into an AGG. Then that AGG is going to be translated into the amino acid arginine. So we call this a missense mutation because it took what should have been a lysine and it changed it into an arginine. So it's going to just put the wrong base, like a misstep in the translation process. So those are our three types of point mutations or SNPs. We have our silent mutation, which results in the same amino acid being placed, even though there is a slight change in the nucleotide sequence. There is a nonsense mutation, which results in a stop codon in the protein levels. That would be an ending in protein synthesis. And then there is a missense mutation, which means that the wrong amino acid is going to be placed at the protein level as a consequence of this particular genetic change. So we just got into the different type of point mutations that we can see, so let's get into the other types of mutations. All right, so here are some of the different types of mutations that BIO1000 wants you to know about. So let's start with the first one, which we have substitutions. We have our standard sequence here. We have both strands, and it reads GA on the top and CT on the bottom. So looking at our first type of mutation is a substitution. So here, in the case of this, what should have been an A, it was substituted for a G. So now the code reads GGGA and the bottom reads CCCT. So that was a substitution of an A and a T for a C and a G. So the next type of mutation is a deletion mutation. So our strand should read GAGA, but now it only reads GGA because there was a deletion of that first adenine base. So then our third mutation is an insertion mutation. So here we've gone from only 4 letters to now we have 5 because there was an insertion of an adenine and a thymine on the opposing strand. So we have inserted an additional base. And then the last mutation that we're going to be going over is an inversion. So here it should be GAGA on the top, but it inverted to be CTCT on the top. So that's an inversion mutation. So something I'm going to kind of mention here is they frequently call insertions and deletions Indel mutations. So the in part of the Indel comes from insertion, and the del part of Indel comes from deletion. So it's a particular type of mutation they like to reference a lot. They call them Indel mutations, so either insertion or deletion mutation. These types of mutations can occur for a variety of reasons, and we're going to go over a few mechanisms of how they come about. All right, so first we're going to get into how indel mutations can occur. So those are the insertion and deletion mutations where either nucleotides are inserted or deleted. So one of the mechanisms that this can occur by is during DNA replication through a process called DNA polymerase slippage. So the first type of slippage that we're going to go over is forward slippage. So this is slippage of the template strand, and that is going to result in a deletion after 2 rounds of replication. So what we have here is slippage in that template strand. So here we have black is our template for our first round of replication, and we see there's kind of a loop in that strand. So there's a portion of the strand that isn't going to be copied by DNA polymerase because it's slipped. The strand is kind of slipped a little bit. So what that's going to result after 1 round of replication is a newly synthesized strand that's a little bit shorter than the template strand should be because it's missing this portion that's kind of looped out of the template strand. So now when we do 2 rounds of replications, now onto our second round of replication, we'll see that this original template strand is going to get pulled apart, and now in this replication it doesn't have any slippage. So now it's going to be copied in its normal way, in its normal length. We see here this blue is our newly synthesized strand in our second round of replication. So we see here this slipped template strand is going to be correctly lined up in our second round of replication and give us a proper DNA helix, the correct length we're looking for. But this synthesized strand in our first round of replication is going to go on and be a template strand in our second round of replication. And because it's shorter than what the original template strand is going to be, it's going to make another strand that's also shorter. So now here we have a DNA helix that is much shorter than what it should have been because of this forward slippage of the original template strand in that first round of replication. So when we have forward slippage or slippage of the template strand, we are going to get a deletion of bases because that portion of the template strand will not be copied into the newly synthesized strand. All right, so that was forward slippage. Let's get into backward slippage. All right, so now we got our backward slippage. So backward slippage is slippage of the newly synthesized strand. So here we can see we have our template strand here in black, and we have our newly synthesized strand in red. And so this newly synthesized strand is going to slip, and it is going to cause the polymerase to think that it didn't copy this part of the DNA that's in the loop. So it's going to recopy it again. And so now this strand, when it goes to be replicated a second time, is going to be longer than it should be, longer than original template strand, because it slipped back while the polymerase was trying to copy it, and it caused that polymerase to recopy parts that it had already put into the newly synthesized strand. So now, when we go to that second round of replication, we're going to have our original template strand in black, and that's going to carry on, and it's going to be the length it's supposed to be. And so it's going to create a newly synthesized strand that is also the correct length, and because there's no slippage that occurred in this particular strand in this round of replication. But then this, what was originally a newly synthesized strand that's going to become a template strand in our second round of replication, because of that slippage, it's now going to be longer than it should be. And so when it goes to replicate, it is going to replicate a newly synthesized strand that is longer than it should be. And so with backward slippage, where you get slippage of the newly synthesized strand, that is going to result in an insertion, because it is going to insert this extra piece of circular that is slipped out of that template strand, and is going to add that when we do a second round of replication. So forward slippage is always slippage of the template strand, and that is going to result in a deletion. And backward slippage is always slippage of the newly synthesized strand, and that is going to result in insertion. So that concludes this video talking about different types of mutations with a focus on indel mutations. In the next video, we're going to go over how other mutations are incurred and the concept of tautomers in tautomeric shifts. So if you want to listen to that, stay tuned for that video to come out. And again, as always, if you have any issues with any of the concepts presented in this or any other video or anything related to biology, please visit biologybean.com, where you can access all of your tutoring needs. And please don't forget to comment, like, and subscribe.